right, let me get situated here. All right. Okay, so I am also going to step away from the light a bit. Thank you very much, Aubrey, for the opportunity and for the Lev Foundation to come here and share with you some of the work that we've been doing over the years, and also to learn from you. So I love coming to meetings because it's a two-way street in what we learn from each speaker. So I'm also in messing with metabolism. I'm going to talk to you today about somatotropic signaling, or growth hormone signaling, and aging. And I'll introduce you to some mouse, the mouse that we use primarily. But first, I think we've learned from, almost every speaker has talked about how aging is the major risk factor for many chronic diseases. And if we can really attack aging with this research, that we are going to help not cure, but we're going to decrease the incidence of many, many age-related diseases. And so I think that's the goal, is to increase this health span. And if we get an increased health span, we're going to get a benefit in increasing longevity, perhaps. So I think about these things as kind of a cascade of events. So we have genetics, we have the environment, we have our lifestyle, and they work with each other. So we know that genetics really determines the responsiveness of individuals to the environment and to lifestyle. And we know that the environment and the lifestyle really drive hormone and metabolic responses. And in turn, these hormone levels and metabolism alter aging processes. And so just a little bit about hormones. We haven't heard a ton about them, a little bit here and there. We know that there's been many examples over the years of how hormones impact aging processes or impact longevity, or factors that we think about that contribute to aging. And so in the 1800s, there was a physiologist that decided to inject himself with testicular extracts from guinea pigs and dogs. And he claimed, and he published, that he had increased vitality, increased strength, both mental and physical. And then many years later, in the 30s, it was discovered that that was testosterone that he was injecting. That was the primary molecule. And they've done lots and lots of studies. They just completed several studies called the T trials. It was 7 different randomized studies in men looking at the effects of testosterone. They didn't find any significant effects on vitality or physical or mental health. So it's not done, that story, but I think it's a work in progress still. And then in 1965, they really started, one of the first studies came out looking at pituitary function and the pituitary hormones, where Arthur Everett in Sydney removed the pituitary gland from rats and found that it really did delay many aging-related pathologies. One of the hormones in the pituitary that became important was growth hormone. And so that's what I'll focus most of the rest of my talk on, and we'll talk a little bit in broader terms and then hone down into growth hormone itself. So just in general, if we think about growth factors, we think about growth and proliferation. And they really are related to longevity through these longevity is related to growth through different pathways, but metabolism was what kind of draws it all together. So if you look at the evolutionary ladder, perhaps, if you want to look at it this way, this is basically a variety of model systems that have been used in aging research for many years. So on the lower end of the ladder, we have organisms that use insulin or insulin-like molecules to drive growth. So they really drive fuel accumulation, and that leads to growth and proliferation, whereas in these higher species, the master regulator of growth is growth hormone, and it's driving insulin-like growth factor to impact somatic growth and also impacting metabolism. Then if you alter the levels by reducing insulin or insulin-like growth factor signaling in these species, or if you alter growth hormone, decrease growth hormone signaling, you're going to get an alteration in metabolism, and these alterations in all of these examples lead to a delay in aging. So just a little bit about growth hormone quickly. It does stimulate the growth of muscle and other tissues, but it also has all these metabolic effects. So it's maybe a misnomer when we call it growth hormone, because it raises blood glucose levels and it decreases fat mass because it induces fat breakdown, basically. But with aging, we know that growth hormone decreases. So after age 25 to 30 in humans, there's a decline in growth hormone that's released. And this also happens in other mammalian systems, including mice. And we know that at the same time, you get a loss in muscle mass and strength, an increase in fat mass, you lose bone mineral density, and insulin sensitivity decreases. And these are all some of these are physical characteristics of what we term as frailty. And then if we think about growth hormone, there's evidence out there perhaps that growth hormone postpones aging, and there's evidence that it's more of an aging accelerator. So I'll go through just a little bit of this. If you go to the internet, perhaps, it shows thousands of clinical trials that growth hormone has positive effects in terms of lifespan and aging. And there's been a few studies, a really small one many, many years ago, where they treated older men with human growth hormone for just 6 weeks. They saw an increase in muscle mass, a decrease in fat mass, so aesthetic effects, but there weren't any functional effects. It didn't alter bone mineral content, it didn't alter strength. And then there's been some studies that they've gone on a bit longer, and they also showed the same thing. So they show an increase in fat, or an increase in lean body mass and a decrease in fat mass, but no change in total strength unless they add a steroid to it. So growth hormone alone doesn't have these the effects on body composition and that turn into a functional change or have a functional effect. And then there's a lot of evidence that suggests that growth hormone is an aging accelerator, both in terms of supraphysiological levels of growth hormone and acromegaly. These individuals die at a faster rate. Their mortality is increased compared to the rest of the population. But they've also shown that growth hormone administration causes all sorts of problems in terms of edema, arthritis, glucose intolerance, diabetes, and cancer. So in humans and in mice, it's been shown that pathological growth hormone really shortens lifespan. So the evidence that growth hormone is pro-aging, more evidence, is that a variety of laboratories have shown that growth hormone secretion pattern is actually linked to longevity. If you delete the receptor, you get or have some sort of a defect in the receptor, you can confer protection from both cancer and diabetes. So Walter Longo's group, working with Jaime, I can't say his name, Aguirre, in 2011, and they've had other subsequent publications. They've shown that these individuals that have a growth hormone receptor mutation have 0 diabetes. So this is the normal population, the amount of diabetes in these individuals, and the blank bar on the right is the individuals that have the growth hormone receptor defect. Same thing for cancer. This is the relatives of the individuals, and then they have very little cancer, if any, in these individuals that have the growth hormone receptor mutation. So this population doesn't necessarily live longer, but they live healthier, even in the face of having higher BMIs. So that brings us to our mouse, the Ames dwarf mouse we've been using for many, many years, looking at the effects of a lack of growth hormone. These animals are growth hormone deficient, and how that impacts various aging processes, including some metabolic effects. So, whoops. These animals, the Ames dwarf animals, are in the blue and the green, females and males. They delay their aging in comparison to the wild types here. So there's an increase in average, the mean lifespan, median lifespan, and maximum lifespan. And they have a gene mutation in a gene that drives differentiation of the pituitary gland. So they're actually missing 3 hormones. But the other hormones, TSH, thyrotropin, and prolactin, have been added back, and you don't see a significant change other than a couple of months change in lifespan. So these animals live 49 to 68% longer than wild-type animals. They're highly stress-resistant, they're highly insulin sensitivity, and kind of in human terms, they're living to 120 and 150 on a regular basis. It's not just the lifespan or that longevity. There's a lot of pieces that go into that, and that is in terms of these longer health spans. So a variety of different systems have been looked at in these animals, a few by our lab, lots by many other labs, to give them credit for this. The immune system, cataracts, collagen, cancer incidence, of course, is quite low. I already mentioned glucose homeostasis is intact. All of these different factors are delayed, very much delayed in their incidence and presence in the Ames dwarf. And we believe that this helps them avoid all of these age-related diseases. And this is just in contrast. These animals are the long-living ones. If you overexpress growth hormone, they become short-living. So these growth hormone transgenics are quite large. They have very, very high plasma levels of growth hormone, and they live 50% shorter. So they live to about 12 months of age when their wild type is living to 2 years of age. The dwarf, in comparison, is their wild type lives 2 years of age, and the dwarfs are living 3 to 4 years of age. So we use these two different extremes in growth hormone to study what's going on in terms of aging and metabolism. So this is just a kind of a little bit of a laundry list about how growth hormone status impacts a variety of different systems that are involved in cellular stress resistance and defense. So the Ames dwarf has an increase in their ability to fight reactive oxygen species, so their antioxidative defense is significantly increased. They have enhanced mitochondrial function, glutathione, all the activities involved in glutathione, which include some DNA methyltransferase enzymes and things. These are all upregulated in these animals. And then in the animal that overexpresses growth hormone, all of these activities are very much suppressed. This is just an example about how growth hormone impacts a variety of different things. I'll show you a few different examples. This is growth hormone status impacts the response to calorie restriction. So this is work that was done by Andrzej Bartke's lab many, many years ago. Again, the dwarf animals are out here on the right of the, on the right in yellow. These are dwarf animals that have been put on calorie restriction, so they live an additional 12% longer when they're calorie restricted. And then the green are the wild type on calorie restriction. So even the dwarf not on calorie restriction pretty much outlive the wild type. So you can add these additional components and they still they still are found to live much longer. If you change another metabolite, methionine, so methionine restriction has been shown for many years in different animal models to extend life. So if you reduce your methionine intake by 80%, you get an extension in life in rats and mice. And there's some human studies going on right now in terms of aging processes. But in our mice, we decided to methionine restrict the animals at a couple of different levels. And we found that it doesn't have any impact at all on average or maximum lifespan in an animal that doesn't have intact growth hormone signaling. So this is one example with the Ames dwarf. Another growth hormone mutant is the growth hormone receptor knockout animal. They have a mutated receptor, so they're not able to respond to that methionine restriction. However, the wild type animals that have intact systems get an extension in lifespan. So that's the black line here with the green arrow. This is the wild type of the growth hormone receptor knockout. You get an extension in lifespan also when you methionine restrict. And then the other example is those growth hormone transgenic animals that only live to 12 months of age. If you reduce methionine by 80% throughout their life, you get a 6-month extension in their life. So they go from 12 to 18 months, which is pretty incredible. And their wild type also has an extension in median and maximum lifespan when they're put on methionine restriction. So the growth hormone status impacts how you respond to various nutrients. Growth hormone status also impacts DNA methylation. So there's been a couple of references to DNA methylation over the last couple of days. We know that when you compare 2-month-old wild type and dwarf and 22-month-old wild type and dwarf, you get a very different pattern in DNA methylation. So the dwarf animals that have very, very ultra low amounts of growth hormone circulating,
they tend to be hypermethylated, so they maintain their methylation pattern throughout life in comparison to the wild type, which are losing methylation. Or if you compare just within the genotype, these are dwarf young and these are dwarf old, and wild type young and wild type old. You see that the dwarf does lose methylation, just like the wild type loses methylation, but it loses it at far fewer places, or these differentially methylated regions, in comparison to the wild type. And this is probably the easiest way to look at that, is that these are wild type animals in blue and dwarf in red. Young wild types start out a little over 60% in their methylation, total methylation, and they drop to about 40% over 20 months. And in the dwarf, they start out at over 70% and they drop to about 50%. So the rate of decline is probably similar between the two, but the dwarf starts out with much more methylation and maintains that stable methylation throughout life. This is another example of growth hormone and its effects on some age-related disease. So growth hormone affects a lot of aspects of liver metabolism, but if you also induce a high-fat diet into these animals, and then we've started taking a look at what happens in this case. Just looking here, this is just some slices out of the liver. This is a wild-type liver on high fat, or an animal that had been on high fat, and the red is oil red O staining, so it indicates fat vesicles. This is a wild type that's on a standard diet, so you see a huge difference in the number of fat vesicles in the one on high fat compared to the one that's on a standard fat diet. Whereas in the dwarf on high fat, we still see those oil red O vesicles, but they're very tiny, so they have much, much smaller vesicles in comparison to the wild type. But they have more than a dwarf on standard diet, but it's much more difficult to see. So the dwarf is really handling that high fat diet very differently, and this has to do with lipid signaling, which Roz Anderson talked about. So we are working with Roz and one of her former postdocs, who's now an assistant professor at Madison, to try to figure out what's going on and how they're handling these lipids and how that impacts aging processes. And the other thing I wanted to tell you is that when you put animals, humans included, mice, when you put them on high fat, you get an increase in all these pro-inflammatory cytokines. And we're not seeing that in the Ames dwarf. So a woman named Crystal Hill, she did a study several years ago, and they looked at high fat diet in the dwarf, and they didn't see any increase in those pro-inflammatory cytokines, either in the circulation or the liver. So again, these animals that have very low growth hormone are handling inflammation or incoming signals, nutrient signals, and the response in inflammation is much different in comparison to the wild type animals that are on, or compared to the wild type and compared to animals that are on standard fat. So this is just another example of how growth hormone status can impact disease. And so we've taken the Ames dwarf mouse, crossed him with an Alzheimer's mouse model. That's a double transgenic for APP-PS1, it's called. And these animals respond, or when you have the cross, the dwarf and the wild type, the cross produces something a little bit different in the brain. So in the cross, the APP-PS1 that are crossed with the dwarf mice, you get lower IGF-1, which kind of makes sense. That's the impact, or the impact of the dwarf gene. You get lower accumulation of the beta amyloid in the brain of these animals, both the A-beta 142 and A-beta 1 through 40. The number of plaques are much smaller in the brains of these animals, and they have lower kind of inflammatory processes. So they have lower astrogliosis and microgliosis, yet they have maintained higher levels of cytokines in the brain of these animals. And so these are just a couple of these are all the different controls that were run, the control animals. Let me see, this is the wild type for the APP, just the plain APP, just the dwarf genotype, or the dwarf wild type, I should say, the dwarf wild type crossed with the transgenic, the dwarf versus the dwarf transgenic animals. So we see very different responses in terms of the load of beta amyloid and how that brain responds to the cross. And then a few other labs have looked at this reduced somatotropic signaling in the dwarf, and they found that it confers, they believe it confers this protection against the beta amyloid accumulation by looking at the microRNAs and HSF1 or heat shock factor 1. So they see these very different responses in animals that are created to have Alzheimer's, artificially, when they're crossed with an animal that has very low growth hormone. And then more recently, a graduate student in my laboratory has been looking at skeletal muscle. So these animals, they're about a third of the size of a wild type, and they live really long, and they have very healthy lifespans, but no one's really looked at muscle function or muscle morphometry. So a student named Matt Johnson has been in the laboratory and has started to look at muscles in these animals. So this is the TA muscle in the leg. This is just a laminin stain of a wild type animal, so it's looking at each myofiber, and this is the dwarf. And so what he's been able to show by counting these fibers, that the number of fibers is, that's a cross-sectional area, I'm sorry. So he's looking at cross-sectional area. So over time, the cross-sectional area decreases with age in the wild type. So these are 4 to 8 months, 10 to 14 months, and 18 plus months that are represented here in the wild type, and then the same ages in the dwarf. And you can see that the number of fibers in the dwarf changes very little over time, whereas you see this decline in wild type. The fibers are 50% smaller in the dwarf in comparison to the controls. And the neat thing about that is when you go and look at grip strength, so we have no idea how strong or weak these animals are. If you look at grip strength in the young, the middle-aged, and the old, the dwarf have greater grip strength, higher grip strength in comparison to the wild type. And these are normalized to body weight. So they tend to be stronger when they're younger and at middle age, and then they kind of catch up with each other a little bit when they're older age. And we don't have as many animals in this particular group at this time. So they seem to have enhanced grip strength that goes along with the number of fibers, the increased number of fibers, regardless of that fiber size. And then for data I don't have, I have but didn't show, we've been also looking at frailty. So these animals tend to maintain that grip strength and a couple of other activities that we measure well into old age in comparison to wild type. And when you look at not just at muscle, but if you look at the whole various physical aspects of the dwarf in terms of frailty, they are much less frail at the same age as wild type, even if you compare it biological age versus chronological age. So overall, we think that growth hormone really does modulate a lot of metabolic responsiveness, whether it's to a diet or some sort of other treatment, in comparison to animals that have high levels of growth hormone or normal levels of growth hormone versus these dwarf animals that have very little growth hormone. So we think from our evidence and evidence in lots of other laboratories that reduced growth hormone actually delays aging processes. And these are just some of the things that we've looked at in our own lab, and then there's lots of other information out there. We think that they have kind of an impaired ability to sense nutrients, at least for methionine and perhaps for a few other things. Because when we add methionine restriction, we don't really see any effect whatsoever in the dwarf. But in the wild type animals and animals that have very high levels of growth hormone, we can reduce that methionine in the diet and have a really strong effect. So they're able to, they may have elevated nutrient sensing, and we think that the dwarf animals have kind of an impaired nutrient sensing, and that leads to this slower progression of aging perhaps, or delayed aging. So back to the very first slide, we think that age is a major risk factor for many different diseases, and that if we can find these interventions that impact aging, it'll be hugely important for the development of many of the chronic diseases. And we think that growth hormone plays a big role in this. So growth hormone, When it's present, there's an increased demand for growth. But in these animals that don't have growth hormone, all of that incoming nutrition has to be utilized for something. And so we believe that it's shifted towards shifted towards cellular protection, cellular defense, so that when these animals are hit with some sort of a insult, that they're better able to handle it in comparison to animals that have normal levels of growth hormone. And growth hormone really, on the internet and in other places, it was really promoted as an anti-aging factor, and we believe exactly the opposite, that it promotes aging, accelerates aging, because it really does suppress antioxidant enzymes. It has impacts on inflammation and many other metabolic effects. And we think that those cause a lot of adverse side effects and perhaps shorten lifespan. So this is just to thank all the individuals in my lab that have, and other people and all the funding sources, and I thank you again for your attention.
Yeah, thanks so much, Holly. That was a great survey of a large number of years of fantastic work that you've done on this topic. Any questions? Over there. Let's go with whoever's got the microphone. Yep.
Yeah, thanks a lot. Can you actually up or down-regulate growth hormone in a tissue or organ-specific manner? Maybe the key is really the action, which is maybe in the thymus it's something positive, in other organs it's negative. And the second question, if you have time, can you modulate growth hormone by nutrition in any way, or supplements?
There's probably supplements that might alter growth hormone levels, but it's really very global, as you mentioned. We're seeing global tissue effects and the system working together, and not just individual tissues. We've looked at individual tissues. We studied the liver for many, many, many years, and which is one of the primary targets of growth hormone in terms of metabolism and not so much growth. But it's a difficult target, right? So because there's receptors everywhere, and there's been some growth hormone growth hormone specific tissue knockouts that a variety of people have made, and they don't see huge changes in lifespan. They see changes within those specific tissues, but not in lifespan in general, because it's really much more of a global effect. Does that answer your question?
Hi, thank you so much for this amazing talk. My question was, is there any trade-off for dwarves? Like, are they paying any price at all in terms of function, such as in reproduction maybe? And also, do you have any maybe evolutionary insights about growth hormone levels?
Yeah, no, that's a great question. Yes, there are trade-offs. So the Ames dwarf in particular the females are sterile. The reason is because they're missing growth, or they're missing prolactin. So mice need prolactin for ovarian function, and these animals don't have that. So if you look at a different animal model of growth hormone deficiency, like the growth hormone receptor knockout, or not growth hormone deficiency, but reduced somatotropic signaling, like the growth hormone receptor knockout animals, they have a receptor defect, so they actually have high growth hormone but can't respond to it. They don't have the defect in thyroid-stimulating hormone or prolactin, and they're not sterile. They reproduce just fine. So these particular animals the females can't reproduce, the males can, and the other hormone, thyroid hormone, they're about 1.5 degrees lower in temperature than the wild-type animals. And this contributes some, but to the aging and longevity, but you can, another individual raise the temperature in the rooms to 30 degrees, to thermal neutrality for mice, and they live shorter, but they still live longer than the wild type.
Okay, yeah. Yeah.
So we work on mitochondrial recycling, and people talk about mitochondria as far as energy production a lot. Mm-hmm. But a lot of the pathways that you see impacted here by growth hormone are also impacted by mitochondrial clearance. Can you talk a little bit about the interplay between mitochondria and growth hormone, and maybe the synthesis of growth hormone?
So we've looked at mitochondria in isolation in these animals and in the growth hormone transgenic animals, but not in terms of. You mean mitochondrial biosynthesis?
Synthesis of the hormone.
The hormone
like
itself?
estrogen is synthesized with the mitochondria. Is growth hormone also there? And might modulating mitochondria be a strategy to reduce growth hormone?
I don't know. That's an interesting question. I don't know that I can really answer that. I just know that we've looked at the mitochondria in these different animals with different growth hormone status, and they're very much affected by the lack of or the overexpression of, and we know that there's plenty of mitochondria that doesn't differ in terms of the numbers, but I don't know about attacking the mitochondria to up or down regulate the amount of growth hormone itself.
Interesting.
Mm-hmm.
All right, thank you so much.
Great.